Welcome to the third part of the genetic lecture that will focus on the genetic change. We will go over the topic briefly because this is like the introduction for the genetic. So the blood group antigen are carried on the protein, glycoprotein, or a glycolipid of the red blood cell, as we discussed on the previous slides. Genetic variation mean the genetic mutation. So there is a change occur on the DNA sequence. So as a result of that, the encoded the carrier protein or the enzyme that will help to synthesize the sugar, the carbohydrate, it will get affected. This is also affect on the antigen expression. So let's read protein, glycoprotein, the genetic variation when it's happened to the protein or glycoprotein. This variation on causes because of the variation on the gene sequencing change on the amino cause the change on the amino acid sequence of the carrier protein. Of the genetic variations occur on the glycolipid or glycoprotein, then the variation on the gene encode the glycosyl transferase, alter the sugar structure attached to the glycolipid or the glycoprotein as a key sequences effect on the antigen expression. So we have two kinds of the mutation. Either the mutation happen on the DNA, the amino acid, the DNA, of course, for both way. The DNA variation affect on the carrier. If it's a protein carrier, then change the sequence. If it's a carbohydrate that encoded by the enzyme glycosyl transferase, that mean it will affect the sugar structure that carry the blood group antigen. What's the impact of this variation? It influences the antigen expression, which can result on altered, weak, missing the antigen expression, or emerging of the new or rare antigen and null phenotype. So if it's the mutation affect on the protein causing the loss of the antigen, or it can create a new or novel antigen. So the patient develop autoantibody because of this missing of this antigen, or the null phenotype. Maybe the mutation just make the gene ineffective, and as a result of that, there is no carrier encoded by this gene because the gene is not working anymore. So there is no product of this gene. This is called it a null phenotype. So the null phenotype, when the carrier is missing not only from the red blood cell, but from all the tissues that express this carrier. Example, a KEL system, a single nucleotide variant on the KEL gene, which is the point mutation, result on the threonine to methionine substitution at the position 193, producing the K and Chilano antigen. So if we have threonine on the position 193, we will get the K capital. If we have methionine instead, then we have the Chilano antigen. Why this variation matter in the blood group? The gene mutation causes the change on the carrier. An altered carrier as a sequences altered the antigen, which develop the autoantibody in response to this change. And these changes are recognized as a new blood group antigen. A new blood group antigen, that means it's not established. This is novel. This is not established before, not documented before. If it's documented, then it's already recognized. Then knowing the genetic basis allow the testing for the antigen. If we don't know what's the mutation on this patient, we cannot test for it. So for example, if we go back to this slide, now we know that this mutation in position 139. If we are in doubt, if I have the patient and I don't know if he has big K or the Shilano antigen, I can make a molecular testing and see if the threonine at the position 193, then this patient have the K, big K. And if he has methionine instead, he has Shilano. And this is when we do molecular phenotyping for some patient when the serological it's not limited, and I want to determine what this patient has at the molecular level. I can do the molecular level, and based on the mutation I will decide if he has the big K or Shilano. But if I don't have this information, I don't know what the mutation and what's the location of the mutation, I can't test for it. And this is exactly what this means. Knowing the genetic basis allow testing for the antigen. This ensures safe transfusion and prevent hemolytic reaction. So, mutation alter the protein, or maybe the carrier, I should say here, alter the carrier. The patient produce the allo antibody, and this is recognized as a new antigen. We can test for it by the genetic testing, and this is lead to the safe transfusion. The gene interaction on the blood group, we have something called suppressed genes. Suppressors, sometimes a blood group antigen are not expressed because other genes suppress their expression. For example, and the famous example in the Lutheran system, the Lutheran antigen and the BCAM, which is the carrier of these antigen called BCAM gene, the encoded gene called a BCAM gene. This BCAM gene encode a Lutheran antigen. The KLF1 gene mutation, if there is a mutation on the KLF1 gene that lead to reduce or suppress the Lutheran antigen expression. So when serologically I test for the Lutheran A or Lutheran B or any Lutheran antigen, I can't detect it, or maybe very weakly detected, and very weakly I can do the elution adsorption to detect it, and this is another story. So it's look like a negative, it's look like a null, it's look like that I don't have the carrier at all on the red blood cell because I miss all the antigen. This we called it in-Lu phenotype. In referred to the inhibition Lutheran phenotype, where the Lutheran antigen are weakly or absent from the red cell. Why? Not because there is a mutation on the BCAM gene. The BCAM gene is normal, but because the mutation happened on the KLF1 gene. And this gene, we called it the suppressor gene. Another point is the epistasis, is when the one gene mask or modify the effect of the another gene at the different locus. It's different from the dominant that we're gonna discuss on the next few slides, which is the inheritance between the allele of the same gene. For example, the ABO system, maybe the easy to understand. The ABO system, if I inherited the A from mom and B from dad, A and B, they are very strong, they are equal they have equal strong, what we called it dominant. So if I inherited these two allele from my parent, then I have the AB phenotype. So epistasis, it's different. It's not because I inherited a dominant allele, but because there is a gene mask or modify the effect of the another gene. Let's say the example. I have H gene, and the normal H gene encode the H antigen, which is the basis of the A and B antigen. So in the red blood cells, I should have the H antigen first, then the A or B antigen come next. But if I don't have the H antigen because of the mutation, then even if I have the normal AB gene, I will still not express the A or B antigen. And this is what happened on the Bombay phenotype. Because there is a mutated H gene, I don't have the H antigen, so there is no H antigen is formed. As a result, A or B and B antigens cannot be expressed even if the persons carry a normal AB gene. Animal culture. So how we name the gene, how we name the mutation, how we, for example, determine the location of the gene, this is responsible of the committee called a Human Genome Organization Gene Nomenclature Committee. The gene name appear always in italic, and if you noticed in my slide, I make sure the name of the gene italics and all in capital letter. For example, AQP1 gene control the Colton blood group system. As you can see here, there is a number on the name of the AQP. What's this number referred to? We'll see. They may also have a number associated with them to distinguish them from other related gene on the same family. So for example, AQP1, there is AQP2, 3, and 4. They are all the same function. They are a member of the family, but as when the first one to be discovered is the AQP1, and it's tend to be a carrier of the Colton blood group system. For example, EQP1, where 1 indicates it is the first identified member of the aquaporin water channel gene family. We will inshallah complete the last part of our lectures on the next video.